Hello, everyone, and welcome to today's webinar. My name is Sherlyn Chen from Boehringer Ingelheim, and I will be your moderator for today's webinar. It is also the last webinar of this year that is organized by the GastroPlus User Group. Today's presentation is titled Application of Cellular Permeability Simulation and the PBPK Models to Capture Significance of Transporter Effects on Dose Linearity. This presentation will be given by Dr. Michael Bolger from Simulations Plus. Professor Bolger is currently Chief Scientist at Simulations Plus, Inc. He earned a BA in Biology and Chemistry from UC San Diego in 1973 and a PhD in Pharmaceutical Chemistry from UCSF in 1978. From 1978 to 1980, he was an NIH postdoctoral fellow in pharmacology at the UC San Diego School of Medicine. From 1980 to 2004, he was a professor of pharmaceutical sciences at the USC School of Pharmacy, Los Angeles, California. From 1987 to 1993, he was a co-founder and a director of medicinal chemistry at the Cosynthesis Inc. He was elected fellow of the American Association for the Advancement of Science in 1996. So that's a short introduction about our presenter. Following the presentation, there will be an opportunity to ask questions. You may either send your question using the questions pane on your control panel, or you may ask your question directly using the hand-raising icon. If you are using a telephone to listen to the call, please be sure to enter the unique audio pin displayed when you join the call, so this enables us to unmute your line so that we can hear you. This webinar will be recorded and it will be available to the user group members in the future. So without further ado, we now begin the webinar presentation. Michael?
All right, well, thank you very much. I appreciate the invitation from the GastroPlus User Group to give this talk. And I think the field of the application of in vitro simulations and in vivo prediction for the influence of efflux transporters has really captured a lot of attention. It's the subject of a lot of active research right now, and we are making some significant progress in this area. So hopefully I can illustrate at least what our role has been and what we're attempting to do with some of the software that we've developed. In the outline here, I'm going to start out with something very basic, which is the partitioning of organic molecules into organic phases from aqueous media. And we need to get this first very basic step, the rate into and out of the organic octanol layer or the cellular membrane, before we can move on to predict permeability or to try to optimize any kind of transporter interactions. So I'm going to compare some of the very early theory that was associated with this process and introduce a newer method that we've developed, which is a structure-based estimation for that those rates of entry into and out of the organic phase or the cellular membrane. We'll talk a little bit about the model assumptions in MembranePlus, and then we'll look at some examples of both lysosomal trapping and the estimation of intracellular Km for digoxin, and then PBPK simulation of the in vivo processes. Now, to start out, partitioning. This is a very, not very old, because it would have been about the time that I was doing my postdoc, actually. Anyway, it's a German article, and they were trying to optimize the bioavailability of the quaternary ammonium analogs which are antagonists of the acetylcholine receptor. And the bioavailability of these types of analogs is quite low because of the quaternary ammonium carrying a permanent positive charge. And so the idea was to see how you could modify the structure and improve the bioavailability. Now, you can see on this diagram that the layer A is an aqueous layer, layer B is an octanol layer, and layer C is a second aqueous layer. And so what they do is fill this little chamber with water, they fill this chamber with water, they fill this one with the concentration of the drug that they're intending to use, and then they put a layer of octanol over the top and do some very gentle stirring. And so what you can see is that the concentration in the A chamber is going to drop as the concentration in the octanol layer increases, and the concentration in the aqueous layer over here also increases. Now the concentrations in the A and the C chambers are going to become equal at equilibrium, but the level of the octanol may vary depending on the partition coefficient of the molecule of interest. And so in this case, we're working with methylene groups from 3 to 9 for this experiment. Now, a few years later, in 1977, Hugo Kubini, saw this work and decided to see about developing a theory for the partitioning rate of the drug into the octanol and out of the octanol, and came up with some equations that related to the partition coefficient, so that he could, in a way, develop a prediction for partition coefficient based on the number of carbons that were added. And so his work was one of the early sort of microscopic membrane permeability predictions. Now, we developed MembranePlus. We started quite a while ago when Vera Lukashova first joined our group, and we had a bit of a hiatus on development, and now it's been released for about a year. This is MembranePlus version 1.00018, and you can see some of the default parameters that we have in here. MembranePlus is able to do exactly this simulation. So this is the 8-carbon method using Kubini's theory to predict the disappearance of the drug from the A compartment, the appearance of the drug in the octanol, and the appearance of the drug into the C compartment in a stepwise fashion. And at the same time, we calculate an apparent permeability for this molecule. Now, if we take that entire set of molecules from C4 up to C11, you can see a progressive increase in the apparent permeability from 1.8 times 10 to the minus 6 to 1.3 times 10 to the minus 5 centimeters per second. Also, there's a certain level of membrane retention. We call it membrane retention, but in this case it's octanol retention, due to the different log P, or partition coefficient. And so by the time we get up to a C11 molecule, we're actually seeing 90% of the drug being retained in the membrane. And so this green line, which represents the rate going into that octanol layer, is quite high in concentration, whereas the A and B plateau at a much lower concentration. The only data that was available in that early paper from the German group was for C8, so we don't really have any validation data for these other concentrations. But I think this is a demonstration of at least the initial step to be able to calculate those velocities in and out of the membrane. Now, why do we want to do this? What's the significance today for rehashing a octanol or a organic solvent partitioning experiment? And I think a lot of people today are very interested in understanding the role that a sink or an absorption sink might play on the process of dissolution, supersaturation, and precipitation. So what I'm showing now is a instrument that's been developed by Sirius. This is in their Inform instrument platform. There's a lot of probes that stick down into this instrument for various experiments. But the key is you're going to have an aqueous layer at the bottom, which is allows you to study the dissolution of a molecule, potential supersaturation, and then there's going to be an organic layer at the top. In their case, they've found that I believe it's decanol is the best solvent to use because it really maintains a nice interface between the aqueous layer and the organic phase. But it does provide a sink during the process of dissolution, supersaturation, and potential precipitation. The results that begin to come out of this can be eventually studied in our program MembranePlus, plus the technology we've developed in DDD+. Now here's some data that was derived by the Orbito project in Europe, and you can see for this undisclosed drug molecule that using the instrument that Sirius developed, we can process concentrations in the aqueous layer, concentrations in the lipid layer. We're not quite sure yet how we're going to get out the potential precipitated material, but these types of experiments are the things that we would like to add to our modeling capabilities for in vitro, so we can translate those mechanistic nucleation precipitation parameters derived by fitting to a system like this with a sink into GastroPlus to get an early prediction of the in vivo precipitation. All right, so if we move on from the Kubini methodology to what we've developed for our structure-based training from CACO2 data that we obtained from absorption systems, we have 22 drugs, we have 44 different experiments. One was at pH 7.4 in the donor compartment, and the other was at pH 6.5 in the donor compartment. And you can see that after we have built this structure-based model to just simply import the molecular structures, that we can get a very good prediction of the acceptor or receiver well in blue or the donor compartment concentrations in red across a series of experiments. And then, of course, we also have the model developed originally by Kubini as an alternative to the structure-based model. This is the equation that we use for the structure-based model, and it's very simple. We have an intercept, and it's a multiple linear regression. We have a coefficient based on the partition coefficient, another coefficient based on the number of rings that would be different than benzene. So it's an indicator for the presence of a ring other than benzene. Another coefficient based on the difference between the hydrogen bond donor hydrogens and the hydrogen bond donor count. Now, if you're not familiar with hydrogen bond donor hydrogens, that would be a situation where an NH would be essentially one hydrogen bond donor. NH2 would be a hydrogen bond donor hydrogen of 2. And so if you've got a differential there, we would add in a term for that. And then the fourth term is just the hydrogen bond acceptor oxygen count. So with these very simple, easily understood linear equations, we've come up with some coefficients that are used in this default model. And using that, I showed before what the correlation between the predicted concentrations, and these are concentrations that span a time range from 0 to 2 hours for this experiment across all of these compounds. This is the apparent permeability observed on the x-axis versus the predicted on the y-axis. You can see it follows the line of identity, and the fitted slope and intercept is pretty close to unity with a R squared of 0.91 or 0.92. If we try to use the same theory with Kubini's model, where we would be optimizing the terms that relate partition coefficient to those velocities into the membrane and coming out of the membrane, we really can't do it for a diverse series of structures. It seemed to work well for his congeneric series of quaternary ammonium cholinergic antagonists, but it really doesn't work well across a series of drug molecules. Now, if we take our default MembranePlus model and load in some molecular structures and then see how well we can fit the actual kinetics of the donor compartment on an A to B experiment, the receiver compartment on an A to B experiment, and the donor compartment on a B to A experiment and the receiver compartment on a B to A experiment, you can see that across a series of molecular structures published by Aki Häkkinen, we actually do a very good prediction of the actual kinetics of those processes in the KEKO2 experiment. The membrane models that we've developed here are for KEKO2 and MDCK in 12, 24, and 96 well. The model contains a bulk aqueous layer, and a diffusion layer, the apical membrane concentration, the apical cytoplasm, basolateral cytoplasm, basolateral membrane concentration, and basolateral aqueous concentrations. In the PAMPA experiment, obviously we're not going to have a cell, so cytoplasm doesn't play a role. And we have both PAMPA, which is considered instantaneous partitioning, and PAMPA, which is diffusive. So we have the apical and basolateral lipid layer. This is a pretty sophisticated calculation that's done for the partitioning. And you can see that the ionization of the molecule is really important. So we're going to not only calculate what the rate of entry and exit for the unionized species, but we're also going to be doing a calculation for the similar entry and exit rates for the cationic and the anionic species as a fraction of these initial neutral species rates. So for furosemide, you can see we've got a difference between the HBDH and HBD of 1. So 4 minus 3 is 1, and that's because of this NH2 group in the molecule. There are 5 hydrogen bond acceptor oxygens, so the sulfonamide, the carboxylic acid, and the furan ring. Each one of these numbers is multiplied by that coefficient to derive the rate of entry into the membrane, and then we calculate the rate coming out of the membrane based on the partition coefficient, either coming from ADMET predictor estimate or your experimental value of partition coefficient. So all of these are the basic processes for passive permeability. I put this slide in a talk I gave a few months ago, and I knew Alex was going to be in the audience, and I just know he loves lots of big equations. I don't expect anybody to, read in these slides, but there's a good reference here that you might want to look at to understand what our theory is going into these partitionings of different ionic species. Okay, the other calculation that we have to make after we've done the full simulation of the drug going from the donor compartment into the membrane, into the cytoplasm, into the basolateral or receiver compartment, whichever direction you're going, and then finally into the receiver compartment, is the apparent permeability. And in all of the typical KEKO experiments that you do in your normal lab, this is the equation that's used. And this is based on the rate of appearance of the drug in the receiver. There's another set of equations that was developed to correct for membrane retention. And so we will give you a value of calculated permeability for both of these processes. All right, so mechanistic overview. What are the things we take into consideration in this cellular simulation? First of all is the apical unstirred water layer boundary and the bulk solvent. So we have diffusion layers that we calculate for the drug in that donor or receiver compartment to account for that unstirred water layer. The unstirred water layer can change depending on the stirring rate or the orbital diameter of your experiment. Then we are calculating the microscopic rates of entry into the membrane and out of the membrane from those theories I just mentioned before for both the apical and the basolateral. We can add in any number of transporters, which are either influx or efflux transporters. So in the future, we anticipate adding models for systems like the hepatocyte cell culture or the expressed transporter cell lines, or possibly even the sandwich culture hepatocyte system, where we can not only include the hepatocyte cells, but also the processes of biliary transport. Once inside the cytoplasm, we calculate the same microscopic rates going into the membrane of the lysosome, coming out of the membrane of the lysosome. We base that on that ionic equilibrium and the pH changes that might be occurring both inside the lysosome and in the cytoplasm. We can add any number of enzymes into the cytosol for estimation of turnover, and then the same thing on the basolateral membrane for the permeability exiting the process. Transporters on the basolateral, of course, because these are polarized cells, that transporter distribution will probably be different on the two membranes. We calculate the pericellular process, which is got a default setting for a typical Keiko membrane, but we know that every single lab has a different tier value and different potential pericellular permeabilities. So if you've done an experiment with a pericellular, you can easily calibrate that process for your cell line and save the membrane physiology for future use. Then we take into consideration the filter permeability, which probably plays a small role, but it does play some role in the overall process of getting the drug into that receiver compartment. Again, the diffusion layer on the basolateral side, potential protein binding can be calculated both on the donor and the receiver. So in this case, apical and basolateral. If you want to add BSA to one and not the other, or add BSA to both, those levels of binding can be taken into consideration as the fraction unbound or by using a Langmuir isotherm. Finally, potential loss to plastic that is nonspecific, and also potential, the evaporation from the donor compartment and sampling processes. Okay, now we're going to move on to enzymes and transporters, and we're using classical Michaelis-Menten kinetics for both metabolic processes, for efflux transporters, and for influx transporters. So these are probably familiar to you if you've used GastroPlus. We have the same kinds of equations in GastroPlus for our PBPK models and for our gut apical and basolateral regions. Lysosome, this is ion trapping, and it's becoming more obvious that the observed plasma concentration time profile for weak bases that have pKa's possibly above 6.5 up to 9 or 10 can be influenced in this way if they're lipophilic. If you have a molecule that gets taken up into the lysosome and at the low pH in that environment, more or less trapped in there, you can not only affect the dwell time of your drug going through the enterocyte, which slows it down and makes Tmax much longer than you would expect, but it will also influence the drug that's available in the hepatocyte for metabolism or efflux into the bile or into the systemic circulation. So you can set up all of the parameters for this in terms of number of lysosomes, lysosomal membrane thickness, volume, and pH. And because we can change the pH, we can do a virtual experiment for any drug that we want to import into MembranePlus to see if it's logical that their lysosomal trapping could be an issue. We can set up a sampling protocol in which we take a sample and either replace it or not replace it. So, we keep track at each point of a sample what the volume change would be and how that might affect concentration. And then we can provide simulation results. As in GastroPlus, we have a location where you can store your observed values. If you've calculated your permeabilities or your membrane retention or your recoveries or mannitol permeabilities, all of these can be stored in the database to be compared with the observed value. Apparent permeability is here, and the apparent permeability is compared to this calculated pericellular permeability so that we can take the total minus the pericellular and calculate the transcellular component. And then we also calculate the permeability with a membrane correction, according to Avdeef's theories. Final receiver concentration, membrane concentration, donor concentration, and membrane retention. So quite a bit of information comes out from a simple run. And we can then monitor the kinetics of the donor and receiver, the cytoplasm, and a number of other parameters. You can also look at the regional percent of the applied amount that resides in the various compartments at the end of the experiment. All right, now we get on to some of the examples. So I want to draw your attention to a few key references that have evolved. Oh, actually, this first section is just a example for the role of lysosomal trapping in CACO2 experiments. And in part, our renewed interest and emphasis in developing the MembranePlus program and releasing it was inspired by some of the work that was done by the group in Finland. Aki Häkkinen, when he was a student with Monkonen, did some really good work. Korjama also is, I'm going to have some references, but they did some very good in vitro data collection as well as cellular simulation modeling. And we could see the value in continuing our development of MembranePlus due to some of their work. This just shows the model that they developed in this paper. They looked at propranolol. I'm going to do this in the presence and absence of baflomycin. Baflomycin is a poison which it inhibits the proton pump from being able to pump protons into the inside of these endosomal vesicles to create a lysosome with lower pH inside. And so if you add baflomycin, you don't have that pH gradient that would allow for lysosomal trapping. So here's propranolol alone with a typical lysosomal pH of 4, and we can see we can fit from Aki's data the donor compartment, the receiver compartment. We can fit the cytosol. Well, there's no concentration from the cytosol from the paper, but you can see what the cytosol concentration would be. But you can also see that the lysosome concentration rises and goes up to a very high level. And this is whether we're going from A to B or from B to A. So the colors change here. The blue is always that basolateral compartment. If we do the experiment again, but this is data derived, so the square points here are the data derived in the presence of bafilomycin. But we're doing the simulations in this case under the same conditions as we did with the control experiment with propranolol, assuming lysosomal pH of 4. If we assume that lysosomal pH of 4, we're getting lots of lysosomal trapping, both A to B and B to A, and we're not fitting the basolateral concentrations very well. We're kind of missing the apical concentrations, and for the B to A experiment, we're really not getting the apical buildup as fast as it should be. But if we just simply change the conditions in MembranePlus to do the simulation with an assumed lysosomal pH of 5.5, now we begin to nicely fit all of those apical and basolateral concentrations for both experiments, plus the degree of lysosomal trapping has dropped by a couple orders of magnitude. So I think you're going to find this useful in a variety of tests for your molecules, where doing the in vitro experiment, granted, is standard operating procedure, and so why would we want to ever simulate? Well, one of the reasons is to be able to do some of these early tests and maybe even do them before you run your in vitro experiments in order to give the people who are setting up the experiment some idea of what concentrations to expect and what kinetics to expect. Now, here's just showing, direct import of molecular structures for propranolol, testosterone, and ibuprofen, and you can see that the default lysosomal accumulation using a pH of 4 in the lysosome is very high with the high pKa base propranolol, essentially nil with testosterone, and actually lysosomal concentrations below the cellular concentrations for ibuprofen. Now we change gears again, and we're going to look at some of the literature that's been developed with regard to both PBPK in vivo simulations and cellular simulations for the fitting of intracellular Km of efflux transporters. And this article by Troutman and Darren Thacker in 2003 looked at and observed this asymmetric effect of PGP on absorptive versus secretory transport. So if we put the drug on the apical side of a KCO2 experiment and look at the apparent permeability, that's going to be different than if we apply the drug to the basolateral side. Now, this differential in efflux ratio has been known for a long time, but they extended it with a concentration dependence calculation so that they could determine what their apparent Km would be. And they found that the Km was 6-fold higher in that absorptive direction. If any take-home message comes out of this talk over the next few slides, I hope I can convince you that when you are trying to derive parameters to put into GastroPlus for modeling the nonlinear dose dependence or significance of an efflux transporter substrate, that the values derived strictly from an in vitro experiment using concentrations in the donor or receiver compartment are not appropriate to predict those concentrations and the Km for that nonlinear in vivo process. In 2007, the group in Finland looked at apparent Km as PGP expression increases. So instead of just doing absorptive and secretory, they had several cell lines with different levels of expression of PGP. They had KCO2 cell lines and also MDCK cell lines with MDR1 overexpression. And they found that the Km value differed significantly depending on the PGP expression. So if PGP in the membrane is low, it has a low expression, you're not getting very much efflux, so intracellular concentrations are relatively high. But if you have a high expression of PGP and you get lots and lots of efflux, then the intracellular concentrations don't build up no matter how much you put on the outside, and so you're going to get a much higher apparent Km with higher PGP expression. In some work that we did with Maria Tubik, who was a student with Peter Longuth, we did the in vivo simulations for tolinolol, which is a substrate of PGP but does not have the complication of any 3A4 or CYP metabolism. They had measured some apparent Kms previously using in vitro experiments at 412 micromolar, and when we tried to reproduce the nonlinear dose dependence for tolinolol in that experiment, we found that that Km would never work correctly, and so we actually had to lower the Km to 0.69 micromolar intracellular Km in order to fit the in vivo data and get the right nonlinear dose dependence. At that time, we hypothesized that the concentration in the membrane, which is where the site, one of the sites for PGP uptake would be important. But I think subsequently that intracellular Km calculation is going to become important. Then a few years later, we updated that with additional simulations for tolinolol, where we included both the OATP1A2 influx and PGP efflux, and the apparent Km from their experiments compared to our now fitted updated value of 6.3 micromolar. Again, shows that the intracellular Km is going to be lower for these substrates. 2010, again, the Finland group, this time with Aki's work, defined a number of different experiments using KCO2 and NBCK overexpressed MDR1 cell lines to correct for the influence of the aqueous boundary layer, the potential membrane permeability changes as pH changes, for PGP expression changes, and for potential lysosomal trapping of quinidine to establish a true intracellular Km. And I'll go into those a little bit more in detail in a second. In 2012, we did a study in conjunction with Bilal Abu Asal, who's now at FDA. He was a student at the University of Louisiana, and this was to do a PBPK model for a sort of discarded Pfizer compound that was a substrate for both 3A4 and PGP. And again, we found that the nonlinear dose dependence, which was much greater than proportional, required a intracellular Km that was lower than the apparent Km observed in vitro. There's some newer studies. Sybil Neuhoff at SimSip has done some work on a semi-mechanistic PBPK model for digoxin, and in that case they used the straight apparent permeability without any correction for intracellular. Some more brand new work that is coming out of Amin Rostami's lab, Matthew Harwood has been working on doing the absolute transporter expression levels, and I think that in terms of true in vitro, in vivo extrapolation, you might want to take a look at Harwood's latest publications for some of the more up-to-date information we can use in that area. So let's go back to Aki Hakinen's work. Here again, they did a cellular simulation in order to derive what the intracellular Km would be from the apparent Kms. And you can see in this column right here the apparent Kms that were observed under different conditions. So if you use set pH 7.4 with quinidine versus pH 6.4, the Km varies quite significantly. If you inhibit the transporter of lysosomal acidification, the Kms vary significantly. If you change the orbital diameter, so normal orbital rotation of a cell culture transwell experiment is about 3 millimeters, if you or 3 centimeters, I think. I'm not that great at the experiments. Anyway, if you reduce it to half the size, then the Kms change. And finally, in the KCO2 experiments at different pHs, you can get different Kms. By fitting all of that data in a cellular simulation, they ended up coming down to a single value of Km that worked across all the experiments. And so this is the key. This is something that you cannot do just by eyeballing your data with those apical and basolateral concentrations. You actually have to do this cellular simulation, and that's one of the reasons why we've developed MembranePlus. Again, this was the study we did with Bilal Abu Asal and Amal Kadumi in University of Louisiana. I thought this was kind of an interesting example. The reason that this is useful, and I don't know, not a lot of people have read this paper, but you can see the marked nonlinearity. If this is the line of identity for a linear dose response, this shows what was observed for the different doses of this UK compound for AUC 0 to T and for Cmax. And this is really a quite significant increase in the exposure, which is probably one of the reasons why they dropped the compound. When we developed the PBPK model for this, we had to use some in vitro values for 3A4, but we did have to fit the Km for the PGP. And these bar graphs are showing at different doses what the fraction absorbed would be in the gray versus the fraction that is metabolized. So at very low doses, where the concentration in the intracellular enterocyte compartment is below the Km for both PGP and 3A4, we see a strong effect of PGP, because if you're below that Km, then PGP is able to do a lot of efflux, and we end up with a very low fraction absorbed. We also, at that point, end up with a low level of metabolism. So in that case, PGP is protecting the molecule from interaction with 3A4, and Darren Thacker's theories about PGP being protective is observed. As we begin to increase the dose and start to saturate PGP, our fraction absorbed goes up and up and up and up, and eventually we get to quite a high fraction absorbed when PGP is completely saturated. But at that point, in this model, it turns out that the 3A4 is not yet saturated, and so we're beginning to see a much higher level of metabolism, primarily by the gut, due to the enhanced access of the drug to the 3A4 in the enterocyte. And so now we're shifting into a region where Les-Bennett's theory of PGP allowing the drug to come back in and move down the enterocyte and come back in and be subject to more metabolism is playing a role. But we would have had to saturate the PGP at that point to observe that. And then eventually we end up with much lower turnover because we're saturating the 3A4, and that's what results in this much greater than proportional nonlinear dose dependence. So if you're interested, take a look at that paper. All right, I got to move along here. Digoxin is a very interesting molecule. It has kind of a moderate partition coefficient, reasonably low solubility, but not very low. It has an increase in solubility in FASF and FESF conditions. The permeability in the A to B direction, measured by absorption systems, is quite low. So this is 5, 6, 7 times 10 to the minus 8 centimeters per second, and in the B to A direction, 7.6 times 10 to the minus 6 centimeters per second. So the B to A ratio is quite high. We can also estimate the passive permeability by taking the average of these two. So we start out with this as our passive permeability. That converts to a human jejunal permeability of 1.4 times 10 to the minus 4, moderate permeability. Now here's a quote from a 2007 article, and it says, although modeling of digoxin pharmacokinetics began 40 years ago, a comprehensive model that explains the roles of transcapillary permeation and tissue binding is still lacking. Well, I don't know. I just wanted to point it out that this is not an easy model to develop. We have a PGP substrate that reaches Cmax 1 to 3 hours after administration. It's excreted predominantly unchanged in the urine. The renal clearance of digoxin is greater than the creatinine clearance, which indicates that there is most definitely tubular secretion by transporters, and there are studies that indicate that it is a substrate for uptake in rat intestine. It is its target is sodium potassium ATPase in skeletal muscle, and so this binding to sodium potassium ATPase actually acts as one of the processes involved in the distribution and the large volume of distribution. This process is slow, and I think the process is responsible for a lot of the distribution kinetics. So if we follow this in the intestine, this just shows our digoxin moving through the lumen, being absorbed passively, and then passively getting into the portal vein. But in addition, we're going to have digoxin in the enterocyte potentially being effluxed by PGP. We're also going to have potential uptake of digoxin by OATP and transport of digoxin through MRP transporters into the systemic circulation. Our model doesn't contain all of these details, but these are things that, you know, if you want to get truly completely mechanistic, you might have to consider. In the liver, the digoxin is coming in through the portal vein and is taken up both passively and also actively via this sodium-dependent multivitamin transporter. I've got the references for all of these, so if you're interested, just email me and I can give you more info. Once inside the hepatocyte, the MRP2 potential and MRP3 processes govern how much goes out in either direction. And then when we get into the kidney, there's uptake by OATP4C1, as well as passive and filtration by fraction unbound and the glomerular filtration rate. And then we've got distribution into the skeletal muscle and binding to sodium potassium ATPase, which is where the antiarrhythmic action occurs. I think what did I do here? There's something, okay. All right, now let's take a look at this Troutman experiment. How do we begin to work with digoxin and understand its intracellular Km? Well, these were experiments that were done in the B to A direction for apparent permeability, the efflux ratio, which is on this axis, and the apparent permeability of the A to B direction. So you can't see it too well on this graph, but I've done some fitting in the next slide. Our first step to develop a good value for the intracellular Km of digoxin is to come up with a slight modification of our default structure-based predictions. So where I earlier in the talk was touting this structure-based calculation as being a panacea for all molecules, it definitely is not in the case of digoxin. Here we have 14 hydrogen bond acceptor oxygens, and in this case the value of minus 0.82587, which is the default C4 value, causes the entry into the membrane and exit from the membrane to be too low, resulting in an apparent permeability prediction that's quite a bit lower than the observed. So we simply take an experiment where the PGP is inhibited by GW918. In this case, you can see these bars are showing the absorptive and secretory permeabilities of digoxin measured by Troutman, and when it's inhibited by the GW compound, they both level out to about the same value of 1.4, 1.5 times 10 to the minus 4 centimeters per second. By changing that C4 value from minus 0.8 to minus 0.4, we diminish the influence of all of these hydrogen bond acceptor oxygens, and we can get a simulated permeability that matches the passive process. So that's our first step. I've done this with a number of our contract study clients, and it is a good way to process this kind of data. Now, if we were to just do the fitting of the in vitro data, you can see that on the left-hand graph, this is fitting to the beta A apparent permeability. As you begin to increase the digoxin concentration and it saturates, we see that permeability drop. The same thing goes for the efflux ratio. As you begin to saturate the PGP, the efflux ratio drops and approaches the level of 1, which is what you would expect for no PGP influence. Those two plots have a half maximal saturation in vitro of around 240, 220 micromolar, which would be about here. If we take a look at the A to B experiment, in this case, just like we've heard before, the secretory versus absorptive process have different apparent permeabilities. And in this case, the Km that's fitted is 2,000 micromolar because it takes a lot higher concentration to saturate when the PGP is actively pushing the drug back out into the apical compartment. Now, the second step in our MembranePlus experiment is to fit the intracellular Km, and so we use the optimizer in MembranePlus across all the records in the database. Now, this involves both the A to B experiments and the B to A experiments, and we're going to be fitting both Km and Vmax. We want to find the right Km, but we can't get, as Km changes, the apparent permeability will change, and so the Vmax has to go along with that. But because we're doing this across a series of concentrations, we can separate those two. If you were trying to do it on one concentration, then it would be an infinitely flat sum of squared surfaces. And in this case, we end up with a fitted intracellular Km of 95 micromolar. We took that Km and went back to some data that we had collected in a in-house contract study with Quotient, who did the studies. And in this case, we could define the exact concentrations that we wanted to run and the number of data points, and we really designed the experiment to get at this idea of PGP efflux. And you can see that we very nicely fit both the apical and basolateral concentrations using the KM that was derived from Troutman's data in 2003. Okay, so the overall GastroPlus PBPK digoxin model we're going to talk about is going to have a permeability limited liver, permeability limited kidney, and a permeability limited muscle, so that we can account for the sodium potassium ATPase binding. These are the intravenous data and also the urinary excretion data, and so those are two hard observed clinical pieces that we have to keep in consideration when you try to fit. And using that PGP KM that was established from the in vitro experiment, plus making some assumptions for the KM of interaction with sodium potassium ATPase, that value came from the literature, and the secretory transporters in the kidney came from the literature, but the Vmaxes need to be adjusted because at this point we really can't do an in vitro, in vivo extrapolation for those Vmax values. You can see we get pretty nice predictions for both 0.5 and 1.5 milligram intravenous, along with a good prediction of the urinary excretion in those two cases. Now, if we go to the PO administration and the influence of particle size, we've got an article that describes 0.25 milligram PO with a small particle and a large particle. And so here's the experiment to calibrate the system with solution dose. And this is steady state data taken at day 8. Then we go on to the PO data with the tablet formulation and a small particle size. This is 6 micron radius. And again, all of the parameters we developed for the intravenous data from a different paper, plus the in vitro experiments, is working nicely to predict the PO. This just shows the multi-day administration. And we can also then predict nicely the decrease in Cmax and AUC due to the increase in particle size for digoxin in this experiment. Okay, I know that was a lot of information in a short period of time, and the summary here is that the classic Kubini method is useful for doing these simulations of drug partitioning into organic layers and membrane layers, but that as far as diverse structures and potential hydrogen bonding considerations, Kubini probably is not going to work for doing good a priori predictions. Our structure-based estimation, I think, is going to work in many of the standard cases, but we still have a relatively small training set of molecular structures for that. And any help that we can get from our partners in improving the data set for this, structure-based development, we greatly appreciate. By studying and using the structure to just simply get the rate of entry into the membrane and out of the membrane, we're not trying to fit all of the other processes. So you'll see a lot of QSAR models that try to predict permeability of CACO2, but then you're talking about entry into the membrane, exit from the membrane, any potential transporters, cytoplasmic diffusion layers, everything else. We just look at the structure-based to define that rate in and out of the membrane, and then we use the simulation to handle everything else. I think the CACO2 or MDCK cellular simulations can be used to estimate that intracellular unbound Km for substrates of efflux transporters, and that's the value that should go into gastro. And that PBPK simulation could then explain the ADME of complex molecules. So I'm going to stop there. Appreciate the attention and the people joining. Our simulations technology team, Jim Mullen and Kay Zito, were the primary developers of MembranePlus once we reactivated it, and Vera Lukachova started the process several years ago. Hai Ying and Jessica are both members in that group and are doing good programming in our biological therapeutics and our additional dosage routes. Marvin Waldman always helps us greatly with theories and a lot of the calculations and processes that we're working on, and we get great support from our computational chem informatics groups and computational groups. So thank you very much.
Okay, thank you very much, Michael, for the great presentation. At this point, we will open up for questions. I saw we have questions coming in. We will go through it in the order they were received. Our first question to Michael: how it will help to elucidate the carbohydrate molecule absorption on plasma membrane protein?
Let's see. I'm having a little trouble viewing the question, but say that again, that we have a plasma membrane. Oh, carbohydrate. Okay.
Yeah, how it will help to elucidate the carbohydrate molecule absorption on plasma membrane protein?
Okay, well, there's a very good question, and it's not something that we have added to the model yet. We definitely have that unstirred water layer diffusion boundary, but we haven't considered that carbohydrate interaction as one of the processes that might influence the absorption. So we just would need some more data, and maybe we'll have to take a look at the literature. But a good question, and no, we haven't considered that yet.
Thank you. The next question: are the data on slide 13 for compounds used to derive the regression model or the compound outside the training set?
So we go back to the slide 13, and so the question would be, are the data available for this
Are the data on this slide for the compound used to derive the regression model, or use the compound outside the training set?
Well, all of the regression model that we developed for this set of data involves all of these compounds. We did not have an external test set in any way, shape, or form when we developed the coefficients for this default structure-based model. Now, subsequently, we have developed a number of simulations for molecules that are outside of this training set, and we use it all the time with novel molecules and contract studies and other things. So if the question is what is the what would be a good validation external test set, I don't have a slide right here, but I could definitely develop something like that.
Okay, thank you. Michael, can you go to slide number 20? There's a question about that slide. What about the protein binding inside the cell cytoplasmic proteins? Can that be modeled as well?
Another very good question, and at the present time in the model that we have, we do not include intracellular protein binding. I think the interaction with some of the biological membranes is is accounted for, but a potential non-specific protein binding term inside the cytoplasm is not included in here. This is something that we can discuss with the team and possibly introduce as a potential term. I don't know if Vera is online or not, but she might have a comment about that.
All right, thank you. There's next question here. Will it work for small molecule mitochondrial membrane modeling?
All right. Okay, another good question. Now, definitely we can, do the calculations for the intracellular concentrations of the small molecules. We have not added a mitochondrial compartment to this model. That would be something that if there is some useful data and if there's some good simulation reasons to add in a mitochondrial compartment or mitochondrial membrane binding, we would certainly be interested to know more about it and think about adding it.
Great, thank you. There's one individual who's requesting the PowerPoint slides. He's asking whether they can see your presentation. I believe the answer is that the webinar is recorded and they will be posted on the website at the Simulation Plus.
Right, that's absolutely correct. Yeah, the recording of the presentation of the webinar will be available, like all of our GastroPlus User Group webinars are available, and the slides can be made available as well. Okay.
And this is the same person mentioned that he's from China, and he says it's late at night, and he cannot get the whole details. He hopes he can get the slides, and he provided his email address.
Okay, well, I greatly appreciate people from different parts of the world attending, and I think that's a great honor to have the attention. Thank you.
Yeah, so that's all the questions we've received so far from the written panel. Let's see if there's anybody with questions here. I think it's a very, very interesting, very informative webinar, Michael, especially the effort.
How about Arvid Negri? You have a question? Do we want to, how about unmute? Arvind, go ahead if you have a question. If you have a microphone, we can hear you. If you're muted at your site, then we won't be able to, but you're unmuted in the attendee list. Okay, maybe Arvind does not have a question.
No, Arvind, right?
Yeah, I was doing Arvind. How about...
he said no, thanks. Yeah, he did the reply.
Oh, all right. How about Prashanti?
Prashanti, the two questions he did put in writing.
Oh, okay. And Raymar?
Raymar.
This is Raymar.
Yeah, go ahead, Raymar.
It's a great seminar.
Yeah, exactly.
Raymar.
I just wanted to say this was really a very comprehensive overview. Thank you, Michael. It was very interesting. I've learned a lot. So no question, just a comment.
Oh, okay, okay. Well, thank you very much, Raymar. Appreciate that.
I was going to say that the software, that the capability to be able to correctly determine the intracellular Km value, which is really unique, very, very, very great, because that will enhance the the accuracy, the reliability, the outcome of the simulation. So
well, you learn a lot about your system when you do these in vitro simulations. And our models in MembranePlus will evolve to include additional systems like the sandwich culture and like the dissolution absorption experiments. And we don't know yet how valuable some of these will be in doing predictions for precipitation and supersaturation, but I think that it is going in the right direction of getting valuable in vitro inputs that can be translated into appropriate in vivo simulation inputs.
That's right, and this is a very complex system. A lot of factors will affect the correct determination of the parameters. So it's not like very simple, straightforward things. Yeah, and more data, of course, with time will be very helpful.
Something that you know, should occur if you're still listening is some laboratories have unique molecular structures and unique targets that they're working on. I know some of the people in Monash University are working on anti-malarial compounds, which have extremely high partition coefficients. And consequently, when they do their permeability experiments, they have to change the CACO2 incubation conditions to include you know, relatively high levels of protein on the apical and basolateral compartments in order to just simply get the drug to come through in some kind of, you know, using them as a sink. Those systems can very easily be calibrated. So run a few standard BCS compounds in that system, measure those kinetics, fit it in your MembranePlus model, so you end up with a novel structure-based calibration for those BCS compounds, and then you can take any new compound you want in that system. It could be a non-standard CACO system, and you will be able to do a prediction for the new compound that can be put into GastroPlus and a human jejunal permeability estimated directly. We do this all the time for absorption systems, CACO2 values.
Yeah, well, we will give another few minutes for more questions. I have one question, Michael, for you. In your talk, one of the examples you mentioned about this compound as the substrate of the efflux pump, and then the A2B and B2A CACO value is significantly different. And what the approach is, you take the average of the two values and consider that as a intrinsic or passive permeability. How good is that approach to assess the passive intrinsic permeability this way?
Well, let's take a look at the numbers. Let's see if I've got the okay. So now we're looking at digoxin, and we're looking at the apparent permeability when we saturate PGP. And it's going down from 2 times 10 to the minus 5 to about 1.2 times 10 to the minus 5. And going in the A to B direction, that permeability, even though the Km is higher, it's going to eventually saturate up here around 1.2 times 10 to the minus 5. So the A to B, the B to A, when the PGP is saturated, should have a passive permeability. If I were to, of 1.2 times 10 to the minus 5. If I were to take the assumption that I used for the digoxin, oops, where am I here? Of the average of the A to B, B to A measured by absorption systems, then I would have come up with 0.38. Now these values measured by absorption systems are different than the values measured by Troutman as an absolute, because these are totally different laboratories working on this process. I'm trying to think if I were to do the calculation of the average for Troutman. I probably should do that. Bottom line here, I think, is that this can be used when you do not have the experiment with the inhibited study. So let's go to MembranePlus on the digoxin and take a look at the inhibited. Here's the inhibited A to B. So the observed A to B, B to A is 1.3 times 10 to the minus 5, 1.48 times 10 to the minus 5, and we're simulating 1.4 times 10 to the minus 5. That's the inhibited. If we go to the low concentration, that would be 2 times 10 to the minus 6 and in Troutman's data. And the B to A at the low concentration is 1 times 10 to the minus 5. So if we take an average of those, what would we come up with? And there's obviously some variability, because if you go to the next concentration, it's 2. So I don't have the number right here, but I do find that it's useful, but if you do have a good experiment where you've run with an inhibitor of PGP, that's the best value to use rather than the average.
Yeah, thank you. I have one more question here. Not actually, the question came in from Lydia. Do you know the correlation factor between the in vitro Keiko model and in vivo studies?
Well, we definitely do for our absorption systems. So we've been very blessed by having a very tight relationship with absorption systems. And so when we are working with, let's see, I think this is the one. When we're working with compounds that have been measured by absorption systems, then we can take these simulated permeabilities that come out of the absorption systems experimental setup. So this is the 24-well system that absorption systems uses. This is the rotation speed. This is the concentration that they run their experiments at. And when we get a permeability determined from that simulation, and this can be for any of your compounds. This does not have to be for BCS compounds. Anything that you import into MembranePlus and run with these conditions for absorption systems, you can come over to Gastro, and then you can use this built-in ABSCA conversion. So we also, in addition to having the in vitro data, we also have the permeabilities that absorption systems has submitted to the FDA for their BCS calibration and classification purposes. And so when you put in a PAP that was simulated by MembranePlus into Gastro and pick ABSCA, then you're going to get a conversion to human, which is pretty accurate. It'll work many very nicely over a wide series of structures.
Thank you, Michael. I saw there's a question from the phone by Fagan Zhang. Fagan, you are unmuted, so you could go ahead and ask your question. Hello? I guess I didn't hear anything from him. Maybe he's already left. Anyway, I do not see any more questions coming in, and I think we, if we If anyone still online and if you have additional questions later on about this presentation, please go to our GastroPlus User Group LinkedIn website where you can post your questions. Your question will be addressed by our members or by Michael or by members in other members in Simulations Plus. So thank you again, Michael, for the presentation and answering questions. For the audience, thank you all for your participation. You may now disconnect from the webinar. Thank you, everyone.
All right, thanks a lot, Shirley. I appreciate you moderating, and thanks to Renee for setting it up.